phototransduction. And this is where we're back in the retina at the back of the eye, and we're looking at a rod. We have two different photoreceptors. We have rods that are more for seeing in dim light and give you kind of different shades of gray kind of vision. And then cones, which this portion is cone shaped. They're more for visual acuity and a lot of light. So we're looking at dim vision, and this is the retinal pigment protein going from the outside in, and this plays an important role that we'll see in the visual cycle. And then notice how the outer segment of this rod kind of has these evaginations here, and that's going to increase surface area for a lot of G protein coupled receptors called rhodopsin, which is a purple pigment. And rhodopsin is the combination of 11-cis retinal plus opsin. So 11-cis retinal is derived from vitamin A, and this is important for night vision because when you see like driving at night and that kind of thing, you're using your rods a lot. And so 11-cis retinal is the aldehyde version of vitamin A, and then opsin is just a protein. And when it's in this 11-cis retinal, it kind of has a bend in it, the structure, and that bend kind of wraps around and fits nicely with the opsin. And so when they're together, there's no light going on. And what you have during that time is what's called the dark current. So if we're in the dark, this channel is open to cations, and that's going to depolarize or lead towards depolarization of the rod. So these EPSPs, these excitatory postsynaptic potentials, are coming in via this dark current. And so if we're completely in the dark, we have sodium primarily and a little bit of calcium added to this coming in. They're both going down their concentration gradients because we know that it's negative inside the cell. And so as they come in, they cause depolarization. And the rod is just a modified neuron, so it acts like a neuron as far as how it releases neurotransmitter. And so in the terminal axon of the rod, you have neurotransmitter. And so when you have this leading towards depolarization, you have calcium come in here, and it'll cause release of that neurotransmitter, and it's inhibitory. So this is an inhibitory neurotransmitter. And what I don't like about this is it's the only time I know of in the body that glutamate, the primary excitatory neurotransmitter, acts as an inhibitory neurotransmitter. So glutamate is the neurotransmitter here, but it acts inhibitory for some reason. So it's going to inhibit this bipolar neuron. When you inhibit this bipolar neuron, you're going to cause less release here. So if you have less release here, this one is an excitatory neurotransmitter, and it also releases glutamate. So glutamate, and this one acts like it normally does as an excitatory neurotransmitter. So when you have less inhibition here, you have more release here. So you have more release of glutamate, which is going to give you a positive effect on the retinal ganglial cells. So when you have that, you're going to have the action potentials going down, telling the brain that you're in the dark. All right, so whenever light comes in, a photon of light, it's going to hit this rhodopsin. When it hits the rhodopsin, it causes the 11-cis retinol to go to all trans retinol. And that just means that everything's on one side, so it makes a nice straight appearance. So if you can imagine, if we had opsin here, and we had 11 cis right now kind of wrapped around it. Whenever light hits, it straightens, and this dissociates away. It doesn't hug it as tight, and that opsin dissociates. And the opsin then goes and activates the G protein. So the G protein, actual G protein has a name, it's called transducin. And transducin A is the alpha subunit right here. And when opsin activates the alpha subunit will disassociate, and it'll go over here to this enzyme. This enzyme is, in the dark, it's cranking out cyclic AMP. The cyclic AMP is being made by a different enzyme. This is called guanylate cyclase, or guanylyl cyclase. So this is the one that's making GTP into cyclic GMP. In the dark, cyclic GMP is going to bind to that channel, and it's going to open it. So what we're doing when a photon hits is this transducin, this alpha subunit from the G protein, is going to go over here and activate phosphodiesterase. Phosphodiesterase is what breaks down cyclic AMP or cyclic GMP. So it's going to break it down from cyclic GMP just to GMP, and that can no longer bind to this channel. And so what happens is this channel closes. This is when a photon hits. It closes the channel, and that's going to lead to a buildup of negative charges inside the cell and lead to what's called hyperpolarization. And you're going to have less release of the neurotransmitter. If you have less release of the inhibitory neurotransmitter, it's going to affect the downstream. So let's go through this part again. A photon hits, and it's going to cause a conformational change in the rhodopsin from 11 cis retinal to all trans. That's going to cause the retinal to break away from the opsin, dissociate, and the opsin will go down here to the G protein, activate the alpha subunit. That's going to go to phosphodiesterase, and it's going to decrease the amount of cyclic AMP inside the rod. The cyclic AMP now can't bind to the sodium-calcium channel to allow those positive ions in. You get a buildup of negative charges within the rod. You're going to have less release of inhibitory neurotransmitter. So this enzyme, guanylyl cyclase or guanylate cyclase, is the enzyme that takes GTP and makes cyclic GMP when we're in the dark. So let's say you get more and more light exposure, you're going to get more and more of this all trans retinal, and eventually, if light keeps coming in, it's going to shift from dim light to more visible light, and you're going to get the cones activated the same way. They get transduced the same way, except they have photopsins instead of rhodopsins, but they'll be activated the same way. So whenever you're in the dark, it's called dark adaptation, where once you've bleached these, this is called bleaching, whenever light hits this and gets it all in the all trans retinal form, is that once you've kind of bleached everything and then you go in the dark, you can't see very well. But once this visual cycle takes place, there's an enzyme. So all this 11 cis retinal has to migrate into the retinal pigment epithelium. It's called epithelium. And so what this does is it has an enzyme that's called cis trans isomerase. If you remember, an isomerase is something that's it's going to just rearrange the atoms. The atoms don't change. So basically, it takes all the all trans retinal. So it's taking the all trans retinal, not the all cis retinal, and so it's going to convert all that all trans retinal that from the light exposure back to 11 cis retinal. And so this is where you have to have vitamin A to have enough of this available. And so if you have a vitamin A deficiency, you can have night blindness because you won't have enough retinal to regenerate the rhodopsin. And so there's also a disease called retinitis pigmentosa, where there's a lot of times there's a mutation in the opsin, and then they lose their vision in their periphery, because rods are primarily in the periphery of the eye. Your peripheral vision is a lot of rods, and then right in the center of the retina at the back you have the fovea centralis, where all the cones are, or most of the cones. And so you kind of get this tunnel vision with that disease. But what's important here about retinal pigment epithelium is as the rhodopsin gets bleached, we end up with a lot of all trans retinal. It has to go back into the retinal pigment epithelium, and then when it's in there, this enzyme acts on it, cis-trans isomerase, that takes the all trans retinal back to 11 cis retinal, and then it can come back and reform the rhodopsin. And so you get over about 20 minutes. It takes about 20 minutes to kind of get all of this back to rhodopsin. And so to get that sensitivity back in your dim vision, it's good to be out in it. You know, the first 5 minutes you get a lot of recovery, but then you're best at about 20 minutes after being in the dark. So if you're a nerd like me and like to look at telescopes, it's nice to go out there for about 20 minutes before you really start looking, and you can see it a lot better. So the retinal ganglial cell, this is actually the axons to this retinal ganglial cell is forming the optic nerve, cranial nerve number 2. So that's going to take all the information to different parts of the brain, including the visual cortex. It's also going to go to the superior colliculus to help you track vision. It's going to go to the suprachiasmatic nucleus and tell your hypothalamus that light's coming in, turn off melatonin production, that kind of thing. So these are forming the axons to the optic nerve. There's other little neurons. These are all neurons, all 3 of these. There are other neurons. One's called horizontal cells, and one's that works here to kind of modify things, and one's called amacrine cells that work here to kind of modify things. But we're not going to go too deep into that. I kind of want you to know more of the vertical system. So light's going to come pass through these layers and activate the rhodopsin in the outer segment here of the rod, and then everything kind of moves back this way and then goes down the optic nerve. Okay. So, any questions about that?